Hi everybody, and welcome back for another instalment of Teach Me in 10, the video series where we ask scientists to be our guest and talk to us about a scientific research area or a scientific concept in less than 10 minutes. We want to make science more accessible for you. Now, in this week's instalment, we invite Alex Almeida to talk to us about the secrets of the human microbiome.
Hello everyone, I'm Alex Almeida. I'm a postdoctoral fellow at EMBL's European Bioinformatics Institute and the Wellcome Sanger Institute. And in this Teach Me in 10 video, I'll be speaking to you about my research, where I'm trying to uncover the hidden secrets of the human gut microbiome. So just so we're all on the same page, when talking about the human gut microbiome, we refer to it as the complex ecosystem that contains thousands of different microbial species. Now, most of them are bacteria and viruses, but they can actually span all domains of life. So you can also find eukaryotes and archaea in there as well. What's interesting about the gut microbiome is that if you actually quantify the number of cells that exist in our intestinal tract of these different organisms, they actually equate more or less the same number of human cells in our body. So we're essentially as much microbe as we are human. Now, the gut microbiome has become quite a hot topic in biomedical research. And this is mostly because in the last decade or so, lots of studies have been implicated the gut microbiome composition with several aspects of human health and disease. So on one hand, the gut microbiome composition seems to influence susceptibility to cancers such as colorectal cancer, also influences the incidence of obesity, autoimmune diseases such as inflammatory bowel disease, and importantly, can also protect us against infection. So the gut microbiome itself can act as a barrier to protect against other microbial species that can be highly pathogenic. Now, in the microbiome field, there are essentially two main questions that we're trying to answer. On one hand is understanding who are these microbes, so what are the individual species that can make up a gut microbiome? And then secondly, understanding what are they actually doing? So do they have any specific function that might tell us why they are linked to specific health or disease phenotypes? In order to answer these questions, there are essentially two main approaches that we can take. So let's say we have a stool sample, so a fecal sample that has been collected from an individual. On one hand, we can try to isolate and grow the individual microbial cells. So let's say these different colored circles represent different bacteria. We can try to grow them in the lab, and this actually allows us to perform further experiments on them, for instance, using mouse models. Now, the problem is that a lot of the species that inhabit our gut microbiome have not yet been grown under laboratory conditions. So this means that we need to use alternative approaches, such as DNA-based methods or sequencing methods that allow us to capture the totality of the diversity, the genes and the genomes within these communities without having to actually grow them in the lab. Now, the DNA is actually quite useful and provides lots of important insights. On one hand, the DNA acts as a unique fingerprint. So it can tell us what are the individual organisms, what are individual species that are there. But secondly, since most of the DNA actually encodes proteins that themselves have a specific function, it can also tell us about the biology and the functional potential of all these different species. So in my line of research, I've been trying to answer the question of how much of the human gut microbiome do we really know? Essentially, what I mean by this is if we use these DNA-based approaches, what are the species that we detect that we actually cannot find and have not yet been grown in the lab? To answer this question, I have analyzed more than 10,000 gut microbiome samples. So these samples were geographically distributed. Most of them were coming from North America and Europe, but we did have a good portion of them coming from Asian countries and also a small subset from Africa and South America. And what I did was I used several computational and bioinformatics approaches to tease out the individual bacterial genomes that were found in these gut microbiome samples. And I was able to identify more than 90,000 bacterial genomes from all these different gut microbiomes. Then I compared these bacterial genomes to a highly curated database of cultured bacteria, and this allowed me to determine how many of the species within those bacterial genomes have not yet been grown in the lab. And the results were quite interesting. So we identified almost 2,000 new bacterial species that we previously did not know existed. So here on the left, I'm showing a phylogenetic tree. So this is a tree where the different tips represent different microbial species, and the colors indicate their culturing status. So If it's in green, means these are the new species that were previously, that still remain uncultured. And then in blue, these are the known cultured species that we also used in our study. And what you can see is that the uncultured species actually span quite a wide range of the bacterial diversity found in the human gut microbiome. In terms of numbers, we have essentially more than tripled the number of species that were found in the human gut. So this was quite remarkable for an environment, for the gut microbiome that has been so well studied for the last few years, especially. Now, beyond looking at the diversity of these different species, we were also interested in understanding what could we say about the functional potential of them. So I looked at the different genes that were found in these different groups, and I looked at which genes were found more prevalent in one group over another. And I detected that genes specifically involved in oxygen tolerance, so genes that are involved in resistance to oxidative stress, were actually less prevalent and abundant in the novel uncultured species. This to us suggests that probably these species are more sensitive to oxygen exposure, and this might be one of the reasons why they have been so difficult to culture. So imagine when you're sampling an individual and collecting a stool sample, these uncultured species that we found here are probably the first ones to die when they're exposed to oxygen. So very specific precautions need to be made to ensure the survival of these species to be able to then grow them in the lab. Now, another thing that we notice is that the uncultured species seem to have a significant geographical bias. So what I mean by this is when we compare the prevalence and abundance of the cultured versus uncultured species, which is what I'm showing here in this plot, we did find that the cultured species seem to be more prevalent than the uncultured ones in green across most regions in the world. However, this was not the case in South America and Africa, where the uncultured species were more prevalent than the cultured ones. What this suggested to us is that probably a lot of the novelty is coming from these two particular geographic regions that are severely underrepresented in current microbiome research. Now, until far, I have exclusively been talking about bacteria, but as I mentioned at the beginning, viruses also make a significant portion of the gut microbiome diversity. In fact, the majority of the viruses that exist in our gut are known as bacteriophages. So these are viruses that can infect bacterial cells. So you have, for instance, here the schematic where you have a bacterial cell in purple, and you have the phages here entering and infecting the bacterial cell. Now, they might look scary, but in reality, these phages are actually very important. And on one hand, they allow to modulate the gut microbiome composition and ensure its correct functioning. So in this particular study, we analyzed about 28,000 human gut microbiome samples from about 28 countries, and we were able to discover more than 140,000 viruses in the human gut. Interestingly, about 50% of them were completely unknown. So this was a work that was led by Luis Camarero Guerrero, so he was a PhD student at the Wellcome Sanger Institute. And what Luis also found is that there were about 280 viral groups, so collection of different viruses that seem to be globally distributed. So they seem to be found across all the different geographic continents that we sampled in this study. In addition, we also described and named a new highly prevalent clade called the gubophage. Now, since this was highly new, and this was really the first time that this clade was formally described, it's largely unknown what it's actually doing. But it does seem that this particular gubophage seems to be highly prevalent. It's one of the most prevalent phages that we have in our gut. So in conclusion, with all these different studies, we have provided a massive expansion to the known bacterial diversity of the human gut microbiome. So before, if you think about the microbial universe, we had really a very tiny window into understanding what was actually there. But now, using these new computational bioinformatics approaches, we have a much better grasp of what are different species or organisms that are present there. This is actually very important because it opens now opportunities to better understand what are the specific organisms and mechanisms that are involved in human health. So now we have these reference points that we can use in future studies to guide further experiments and mechanistic understanding. So to conclude, I'll just like to thank the different people who have contributed to this work. So my team, led by Rob Finn at EMBL-EBI, but also all our collaborators, both in the UK and abroad. In case you have any specific questions or interest in this work and want to discuss further, feel free to reach out to me either by email or on Twitter. I put the contact information there on the bottom right. So I hope you enjoyed this video and thank you all for joining.
A huge thank you to Alex for joining us here at Teach Me in 10. If you were interested in Alex's presentation and you want to learn more, please check out the resources in the video description below. We'll be back next week for another instalment of Teach Me in 10. See you then.